Today on Flippin Science, we're talking about turning genes into proteins. Thanks to Sam Moyle for producing this PowerPoint. So the science understandings we're going to look at: protein synthesis involves transcription of a gene into messenger RNA, mRNA, and translation of mRNA into an amino acid sequence of the ribosomes. In eukaryotic cells, transcription occurs in the nucleus. Describe and illustrate the role of DNA, mRNA, transfer RNA, and ribosomal RNA in transcription and translation. Describe the relationship between DNA codons, RNA codons, anticodons, and amino acids. Distinguish between the coding and template strands of DNA, and recognize that DNA is directional and is read from 5' to 3'. Genetic code is a sequence of A, C, T's, and G's. So A, T, G, C, T, A, H, E, G, and so on. So it could be like that, and it could be hundreds of letters long. So coding for one gene, you could have hundreds of letters making up that code on DNA. And it comes in groups of 3. So we have 3 letters together. So AAT, for example, that would be a code that would code for something, a specific amino acid later on. On DNA, we call them triplets, and then they're transcribed into mRNA, and then we call them codons. So that's a bit of an issue with the language there. A codon codes for a particular amino acid. Amino acids are what makes up proteins. So proteins are made up of lots of amino acids joined together. We have 20 amino acids that we use to make up all of our proteins. Amino acids are coded for by codons on mRNA. But we also have starts and stops, because that tells you when to start coding and when to stop coding. So when you start making your strand of protein and when to stop making your strand of protein. We'll talk about the difference between DNA and RNA shortly. Let's get complicated. You need 3 letters to code for an amino acid. So these are codons, these are on RNA. So for example, if you had a codon on your mRNA coming out that was UUU, that would say phenylalanine is what I need. If you had CCC, it would say proline, that's what I need. If you had AUU, that'd be isoleucine, I need you now. So there's 20 amino acids, and you'll see them in the table here. You need 3 letters to code for an amino acid, and you'll see there's some redundancy for some of the amino acids. So some of them, so we've got at least 2 codes. Here we have 6 codes for leucine. So there's some redundancy built in. And we've got starts and stops. So AUG, that's methionine, that means start. And then we've got some stop ones, UAA, UAG, UGA, they mean stop. So they mean stop making my protein now. So the way we show this is often in a table. We can also have little diagrams like this as well. So these are codons on mRNA that tell you which amino acid that you need to grab to join together to make your protein. So if we go back to our recipe analogy, the codons might be individual components of the recipe. So a cup of flour, a cup of sugar, and the method of putting them together. So here we have our recipe. This is our gene, remember from our analogy. If we read through our recipe, we can have things like what we need to make it. So we need this, we need this, we need this, we need this. So these are our parts, and then we need to put our bits together. So to start doing this, then we need to do these 3 things, then we need to do those 4 things, and then at the end we need to stop. So this is an analogy for what's happening when we're making a protein from DNA. The way we make a protein, it starts with DNA, we turn that DNA into RNA, and then we turn that RNA into a protein. When I say turn into, we're making copies of, and then we are reading our copy to make our final product. So there's 2 big processes here, transcription and translation. And both of them involve RNA, and RNA is different from DNA. And that's what we're going to look at down here. Now we have this mRNA and we've got tRNA, but the key things are the RNA, which is a different molecule than DNA. It's only slightly different. It's still a nucleic acids, that's the NA bit, but there's some differences. The sugar is ribose instead of deoxyribose. So in DNA, it's deoxyribose nucleic acid. In RNA, it's ribonucleic acid or ribose nucleic acid. RNA is only made up of a single strand, whereas DNA is double-stranded. It's a double helix. RNA isn't. It's a single helix. RNA has a U instead of a T. So wherever we would normally see T in DNA, we see a U in RNA. So adenine always pairs with U in RNA, not T. And RNA can move. It's not just stuck in the nucleus, whereas DNA is normally stuck in the nucleus in the cell, unless the cell is dividing. RNA can move out of the nucleus, and that's what the M in messenger RNA means. It's going from the nucleus out to a ribosome outside the nucleus. So here's RNA, single strand. DNA, double-stranded. Our sugar is ribose. Our sugar over here in DNA is deoxyribose. See that it's lost an oxygen. So that's the deoxy bit, it's lost an oxygen, so it's deoxygenated. In DNA we have thymine, in RNA we have uracil. So there are key differences between RNA and DNA. And RNA, like I said, it can move. It can go outside of the nucleus, whereas DNA is stuck inside the nucleus. Time for another analogy. Our DNA we want to protect. It's our good copy of the instructions to make our protein. So if you're on a building site, you have a blueprint that stays safe in an office in a building site, so that not everyone can go and grab it and get the grubbing mitt and tear it up and throw it in the bin accidentally. But you need to have a copy of the information in order to make your building. So you photocopy and you get a copy of the blueprint to tell you what you need to do to make your final product. That way you can take it out away from the office and you can take it to the building site, and if it gets damaged or thrown in the bin or torn up, it doesn't really matter too much because you've still got that good copy you can go back to. So that's what mRNA is. mRNA is our working copy that we take from DNA to make our final product outside of the nucleus, so somewhere where it's not protected. So let's talk about transcription. Transcription is this copying process where we're taking our blueprints and we're taking a copy of that so then we can take it out of the office and onto the building site so we can make our final product, our building. So we're making a complementary section of a gene on an mRNA molecule. And that mRNA molecule can then go outside and get turned into a protein. And then that mRNA molecule can get taken out of the nucleus and be read in order to make our final product, our protein. Back to the recipe analogy, the recipe book's in the library, but the library isn't going to let you borrow it because you've lost your card. So instead you might take a photo, or you might write down the recipe, or you might even photocopy the recipe so you can take it home and then use it. So transcription is this process where we're copying our recipe and we're going to use it to make our final product, our protein in this case, outside of the nucleus. So again, we've got a crash course video. I'll put that up on DMAT for you in class. So let's talk about the step after transcription. So we've got our working copy. We then need to read off the copy and turn that into our final product, our protein. So mRNA leaves the nucleus, it goes to the cytoplasm, and it finds its way to a ribosome. Now ribosomes, you should know, these are organelles that make proteins. They're made of a particular type of RNA called ribosomal RNA, or rRNA, little rRNA, and also some proteins as well. So that's what a ribosome is. Its job is to make proteins from the instructions that it gets from the mRNA. This process is called translation. It reads off the codons, so about 3 little codes on our mRNA, and it grabs amino acids as told by those codons and joins them together, taking the ingredients and putting them together to make your final product. So in this case, it's a protein. It gets a bit more complicated. To get the amino acids to the ribosome, we have another type of RNA called TRNA, transfer RNA. We'll look at what that looks like in a second. So let's look at some pictures of these processes. In transcription, we are making our working copy of the DNA on an mRNA molecule. The DNA unzips, an enzyme called RNA polymerase comes in. It makes a copy of the DNA on RNA, mRNA. Then that mRNA makes its way out of the nucleus. It makes its way to a ribosome. This is our ribosome here. Then our 3-letter codons start getting read off, and our amino acids are brought in at the ribosome and get joined together based on our 3-letter codes as we go through. So as the ribosome goes along the mRNA, we get our amino acids being dropped off by our tRNA molecules. Let's talk about tRNAs. So for each codon, we have a specific tRNA. A tRNA is about 80 nucleotides long, and it's folded into a shape. We call it a cloverleaf shape because it kind of looks a bit like a cloverleaf. They've got these little bits hanging off of it. At one end, we have exposed triplets, and we call this an anticodon because it's the opposite of a codon. So if our anticodon is ACC, our codon would be the opposite of that. A bonds with C, C bonds with A, C bonds with A. So it would be CAA. That would be our codon over here. So it's the opposite of a codon, so it's an anticodon. Now at the other end, we have our amino acids. So in this case, it's tryptophan. So tryptophan is being dropped off by this tRNA with the anticodon ACC, which would have a codon on the mRNA, which would be CAA. So each codon on mRNA has an anticodon on a tRNA. Attached to the tRNA, there's amino acids, and that codon is saying, please bring in the amino acid that I need, tRNA, and join it together at the ribosome so I get a sequence of amino acids being joined together. So as mRNA gets translated, the tRNA brings in the amino acid, and then the next one, and then the next one, and the next one, until you get to the end of your mRNA. So until you get to your stop codon. The amino acids are joined together by an enzyme to make a peptide link. So it's a polypeptide. So we get lots of amino acids joined together to make a polypeptide. Another word for polypeptide is a protein. Now, they use tRNA molecules. They go back out of the ribosome. They pick up another amino acid that's specific to their anticodon, and then they can be used again. So let's have a look at this again. So here we've got transcription. We've got our mRNA being produced from our DNA. Then our mRNA makes its way to a ribosome. At the ribosome, we've got our 3-letter codons that are specific to a tRNA molecule with the anticodon compared to that codon. Those amino acids are attached to the tRNAs that are specific to that anticodon. And as the tRNAs come in, they plop in, they drop off their amino acid, an enzyme joins those amino acids together, then the tRNAs go out. Then the ribosome moves on to the next codon, and the process repeats itself until we get to a stop codon at the end. Here's an animation showing that. So we have an enzyme that opens up the DNA, and we're starting to get our mRNA being produced here. So we can see instead of having A's with T's, we have A's with U's now. So here's our mRNA molecule that's being produced. This is our gene, our DNA sequence. So we're making our mRNA until we get to our stop codon at the end. Right, now we take the mRNA that goes into the cytoplasm. Now we have our ribosome coming in, so this is our ribosome here. And now we start getting our tRNAs bringing in. Our first amino acid is always with methionine because this is our start. New tRNAs come in, here are our anticodons, here are our codons, and the amino acids are joined together. So the joint, here's tryptophan that we had before. So they're being joined together until we get to a stop. Once we get to a stop, the ribosome disassembles, and then we've got our sequence of amino acids joined together. And then we have this folding process that occurs, and we're going to talk about that shortly. And this is where the fun questions come in. If we give you one of these 3-letter codes, you should be able to tell us what the other aspects of that code would be. So if I gave you GGA as a codon, you should be able to tell me what the DNA would have been that would produce GGA. So the opposite of G is C, so G always bonds with C, and the opposite of A is always T. So we would have GGA would code for on DNA, so this is our triplet on DNA, that would be CCT. The anticodon would have to be the opposite of our codon. So the opposite of G would be C, the opposite of G would be C, but because this is RNA now, rather than having T, we would have a U. And in the end, that codes for the amino acid of glycine. So this is the fun question stuff that we can do on this. So if we give you one of these, we should be able to figure out the other ones involved. Last bit, we need to know the difference between coding and template strands on DNA. So DNA is made up of 2 strands. The template strand is the most important one in terms of making the mRNA because it's the bit that is read and turned into the mRNA molecule. It is the template to make the mRNA. So if we have a look at our picture down here, we have our DNA. One side of it is being read and turned into the mRNA. That's our template strand. The coding strand is opposite to that. So it's the strand on the other side that isn't being turned into mRNA. What is interesting about the coding strand is it's identical to the sequence that you get on mRNA because these are both complementary. So this is complementary to this, and the mRNA is complementary as well. But you get your T's on the DNA are replaced with U's on the mRNA. So that's the difference between a coding and the template strand. The template strand is used to make the mRNA. The coding strand is the opposite of the template strand. A new addition to the curriculum is talking about how DNA is directional and is read from the 5' to the 3' end of the DNA. Basically, DNA is arranged, one strand goes in one direction, the other strand goes in the other direction. And it's about how the bonds are forming in the backbone. One end is called the 5' end, the other end is called the 3' end. And the key thing you need to remember is that DNA is read from 5' to 3'. So the direction is always 5' to 3'. So that's just another thing that you need to remember. So summarizing there, the section of DNA is copied to make a working copy. It can go out of the nucleus, and we call that process transcription. The copy, which is the mRNA strand, goes to ribosomes where it's read, and the instructions are there to make a new protein, and we call that translation. Each codon is matched by an anticodon on tRNA. tRNA has a specific amino acid attached to it based on the anticodon present on the tRNA. As the mRNA is read, multiple tRNAs write, laying down a pattern of amino acids to make up the protein. and the DNA is read from 5' to 3' when we're doing that transcription process. So the science understands we looked at protein synthesis involving transcription from DNA into mRNA and translation of mRNA into an amino acid sequence at ribosomes. You need to be able to talk about what DNA, mRNA, TRNA, and ribosomal RNA are involved in terms of transcription and translation. Talk about how DNA codons, RNA codons, anticodons, and amino acids are related. Talk about the coding and template strands of DNA, and recognize that DNA is read from 5' to 3' ends. That's it for the today. See ya.